here we are. So first of all, thank you for the audience and our speaker of today, Sujit Datta, to be here to this population dynamics virtual seminar. It's actually the last of the year, and we are gonna to start next year with a new series. So I will send a mail hopefully soon, probably end of December, regarding the new list of speakers. I'll share my screen. Hopefully you can see it full screen. I hope so. So, as I told you, the speaker of today is Sujit Datta from recently moved to Caltech, USA. And the title of the talk is The Wisdom of the Crowd, Watching Microbial Collectives Reshape Themselves.
Sorry, Marco, I should have. Yeah, I just switched up the title. Whoops.
Totally fine. I mean, so the title will be said by Sujit after. And before starting, I would like to remind that we have also, I mean, personally I have, but we were working in the same field, X and now Blue Sky social that regards theoretical biophysics, and we are looking for collaborators. So we have a podcast, we summarize new papers, and we have also we publish in opening position and so on. So please, if you would like to be collaborators, write us something. So regarding the speaker of today, so he studied physics at University of Pennsylvania, and then did a PhD at Harvard University, postdoc at Caltech in chemical engineering, and assistant and associate professor at Princeton University, and now he got a position at Caltech since this year. He's interested in transport processes of soft and living systems, and he studied these topics by combining microscopy, microfluidics, material science, biological characterization with theoretical and computational modeling. And the goal is to shed light on behavior of complex fluids and microbial systems in complex environments. So with this one, I would like to Sujit leave possibility to share the screen. I think I stopped my screen sharing, so actually you can share it and the stage is totally yours. Thank you.
Let's see. Okay, can you see my screen okay?
Yes, we can see it full screen.
Okay, excellent. Well, first of all, thank you, Marco, for that introduction, and thank you for having me here. I'm really it's a great honor to be a part of this seminar series, and I appreciate having this opportunity to share some of our group's research with all of you. As Marco mentioned, one of the things that, my group, broadly speaking, studies transport processes across different kinds of soft and living systems. And one big thrust of our work has been studying the dynamics of microbial life, especially in complex spaces. And so today, I'll tell you a little bit about the work that we've been doing over the past few years. I wanted to give you all a brief overview, kind of a broad overview of some of the things we've worked on in the past few years. And of course, if you have any questions or comments, feel free to, just chime in over the course of the talk. It's more fun that way. And I look forward to interacting with many more of you in the future. So, first, maybe just to give you a sense for why we care about what we care about. Ultimately, one of the reasons why I'm interested in microbial life and the dynamics of microbial life is because, well, life on this planet wouldn't exist if it wasn't for microbes, right? Microbes play important roles in biogeochemical cycling. They play important, have important influences on our health, both beneficial and harmful. These are all things that you all know much better than I do, especially as collectives, as populations. And so when we look at them in the natural world, right, they typically exist in these complex environments, like in soils and sediments, where they help sustain plant growth and perform important biogeochemical cycling, in the tissues and gels of our bodies. And so, microbial populations are often found in these highly complex, messy natural settings with, structural disorder and crowding, various kinds of chemical and mechanical heterogeneities, and so on. right? And so this is the world we inhabit and we find them in. Now, typically in the lab, right, we encounter these microbes in much more idealized settings, right? We study them in bulk liquids, in test tubes, or on flat surfaces, in petri dishes, right? And these studies give us incredibly important insights into how microbes behave, but they don't recapitulate many of these complexities inherent in natural life. And bridging the gap between these two extremes is what we try to focus on in our research, right? And what we do is we try to, oh, I gave this away, whoops. One challenge that we encounter when we want to bridge the gap between these two extremes is just the recognition that many of the complex media that microbes inhabit in the natural world, when it comes to the subsurface, soils and sediments and so on, or the tissues and gels of our bodies, is that these environments are opaque, right? We can't see what's going on inside of them. And so what we try to do in my group is we try to overcome this challenge, inspired by this lovely quote by the great American baseball player and philosopher of life, Yogi Berra, who once said, you can observe a lot just by watching. So what we do is we try to develop experimental model systems that can recapitulate many of the complexities inherent in nature in systematic ways, structural disorder, crowding and confinement, various kinds of heterogeneities, in media that have controlled properties, and that we can tune these complexities in various ways. But best of all, they're completely transparent. And so we can spy on what microbes are doing inside these complex 3D spaces using confocal microscopy. And then, inspired by what we see in our experiments, we try to develop mathematical models to generalize our findings more broadly. So this is the approach that we've taken in my group in many different areas. And ultimately, in the area of microbes, we've been trying to address this very simple question: how does bacterial behavior change when they inhabit these more complex spaces? And so we've worked on this topic for many years. Some of our papers are shown here. These are the people who made this work possible. And in particular, today, I'll tell you about work that was started and led by a former postdoc in my group, Tapa Bhattacharjee, who's now on the faculty at the National Center for Biological Sciences in Bangalore, India. We also had important contributions to the experiments from Konane Bey, who was another former postdoc in the group and is now in the faculty at CU Boulder. Jena Ott was a graduate student in the group who contributed to the experiments as well as the simulations. She's now a postdoc at the University of Delaware. Anna Hancock is currently a graduate student in the group who's continuing to contribute to these experiments. And she worked with Hao Ni Lu, who was an undergraduate in the group and is now a graduate student at MIT. We've been also, some of the experiments that I'll mention at the end of my talk were performed by Alejandro Martinez Calvo, Carolina Trinado Yuste, and Mira Ramaswamy, three postdocs in my group. And we've also developed some simulations and theory that combined with our experiments. And the simulations were started by a former PhD student in my group, Daniel Amchin, who's now a postdoc at Johnson & Johnson, and Alejandro Martinez Calvo and Carolina Trinado used to continue to push that along. And so I'll just jump right into today's story. Today's story begins with E. coli, which I'm sure many of you know and love. It's a nice model microorganism. My lab studies many other species of bacteria, but I'll just focus on E. coli today. And we know that E. coli are motile. They have this wonderful capability to self-propel and swim through fluids through what's known as run and tumble motility, right? This is something that you read about in the textbooks. And we now know, thanks to the pioneering work of Howard Berg and many others over decades, that E. coli swim through bulk liquids using runs and tumbles. And so the way this works is very simple. These bacteria are like little submarines. They're 2 micrometers long, half a micron in diameter, and they have these appendages, flagella, on their surfaces that are attached to the surface with a motor that spins them around. And when they all spin synchronously, they form a bundle that twirls and pushes the cell along a constant straight line path through fluid at a constant speed for around 2 seconds on average. And that's known as a run. And then one or more of the flagella spins in the wrong direction, which causes this bundle to splay out. And that hydrodynamic force causes the cell body to reorient. And then very quickly, the flagella come back together and push the cell along another run in a different direction. And that reorientation process is known as a tumble, and it's effectively instantaneous. It lasts on average 100 milliseconds, which is over an order of magnitude shorter than the duration of a run. And so the cell continues to zip along these runs and tumbles in different directions in a bulk fluid. And so it's doing a random walk, right? And so what's nice about this is you can use the diffusion equation to quantitatively describe how these cells spread over large length and time scales in bulk fluids with an effective active diffusion coefficient given by the product of the run speed and the run length, both of which are just set by the internal biochemical circuitry of the cell, right? And this is a beautifully elegant picture that's been validated in many studies of microbes swimming in bulk fluids over the years, right? And the question that we had was very simply, well, many bacteria don't live in bulk fluids. They live in more crowded, confined settings, right? Like the tissues and gels of our bodies or soils and sediments. How does this picture change when these bacteria move in more crowded spaces, right? And so to address this question, we came up with a very simple experimental platform. And this is the experimental platform that underlies all the experiments that I'll tell you about today.
We have a question in the chat
Yes.
actually, so probably
Okay,
something that
perfect. Go ahead.
anticipates later. Can these bacteria sense nutrients and not do random walk? So probably a
Perfect
direction.
question. Yes, absolutely. And I will tell you more about that in a few slides. Thank you for that question. And if I haven't fully answered it by the end of the talk, feel free to ask again, but I'll get exactly to that in the next few slides. For now, we'll just treat the idealized case where there are no gradients, everything is homogeneous, there's nutrients everywhere, so they are living in chicken soup. And we'll explore the role of gradients very soon. And so to study even just what happens in bulk uniform cases where there's nutrients everywhere, no gradients, but they're in crowded spaces, we came up with this experimental platform, which is essentially like a ball pit for the cells. Okay, so this is
I think I have to interrupt you again because there are
no
probably more
problem.
questions
Thank you.
coming.
This is great.
Ruma, would you like to speak out of a microphone, your question? Maybe will take a while. I think we can go on and Ruma will write it or open the microphone later. Sorry, please.
No worries. Okay. So to study how these cells move in crowded spaces, we came up with these hydrogel ball pits. And so this is like our model of a 3D tissue or a soil, right, with controllable properties. What we do is we take hydrogel grains. So hydrogels are cross-linked networks of polymers that really like water. They're biocompatible. And what we do is we take granules that are individually internally cross-linked networks of these hydrogels. If you take one of these dry granules and disperse it in whatever liquid cell culture medium you want to use with the right nutrients and osmolytes for the cells, then the granule swells up. It absorbs that liquid and those nutrients and osmolytes until it can't swell anymore because of the elasticity of that network. And so you can imagine if you disperse enough of these dry granules into your liquid, then they'll swell up and they'll form a jammed packing, right? Just like a ball pit for the cells, right? If you add more of these dry granules, then you get a tighter packing where the pores between the granules are smaller. If you have less of the granules, then you have a looser packing where the pores are bigger. And so just by tuning how much of these hydrogels you use when you make these packings, you can tune very precisely how tight the packing is and how confined the cells would be inside the pore space. Marco, I don't know if you had a.
Yeah, we have a question here actually, but I think it's more a misunderstanding or a clarification. So Ruma is
That's okay.
asking, why do you take porous media for random walk model? Yeah, I don't think it's actually connected to the model itself, but
Yeah. So the run and tumble model is how the paradigm of how people think about how E. coli and many other related paratrichously flagellated bacteria move through bulk fluids. And so this has nothing to do with porous media yet. Just in bulk fluids, a cell does a run in one direction and then a run in another direction and so on. And so the steps of those runs are like the steps of a random walk. And so people have shown that over large length and time scales, length scales longer than the scale of a single run, time scales longer than the duration of a run, you can treat that as a random walk and therefore use a diffusion equation. That's just in bulk fluid. And the scientific question that we want to address is how is that picture now changed when these cells are inside a 3D porous medium? I hope I answered that question.
thanks.
Yeah. And so this is the experimental platform we came up with. And so what's nice about this is we can make these 3D porous media that are like models of tissues or soils, right? We can tune their structures, how tightly confined the cells would be, or how loose the pores are. We can tune the mechanical properties. We can tune the chemical properties. So this gives us a really nice way of changing the environment of the cells. The individual hydrogel grains are internally microporous. The internal mesh size is on the order of 100 nanometers. We've measured that. And so what's nice about this also is that, the cells, they can't go into the hydrogel grains. They have to go around them through the pores between the grains. But for oxygen or the small molecules that the cells use for metabolism or to signal to each other, which are much smaller than that mesh size, they don't even see this packing. They can diffuse right through unimpeded. And so this platform gives us a way of studying what the influence of physical confinement is on these cells without altering their chemical environment. And so we can, for example, map out the 3D pore space structure. Here we just diffuse fluorescent tracers throughout the pore space. You can see that the pores are irregular. They have different shapes and sizes, right? And so we use mean square displacement measurements by tracking the individual tracers to get a measure of this tightest pore dimension that these cells would feel when they're in this pore space. And so here are just four measurements of four pore size distributions in four different packings. And so the y-axis is logarithmic. It shows the complementary cumulative distribution function. And you can see in blue, this is the loosest packing where you have pores between 1 and 14 microns in size. As you make the packings tighter and tighter, the pores get smaller and smaller and smaller, right? And so we can tune these pore sizes over a broad range. And I also want to just draw your attention to these numbers in the top right of this plot here, right? These numbers show the mass fraction of the hydrogel polymer that we use to make these packings. You'll notice it's always less than 1%. These packings, even though they're 3D solid packings, are made of over 99% water, right? The reason why they form solid 3D packings is because the hydrogel is so highly swollen. And so because they're made of over 99% water, they're completely transparent. And so we can watch what the cells are doing in the 3D pore space, right, just by watching them using confocal microscopy. And so that's what we did. When we first came up with this experimental platform, Tapa took fluorescent E. coli. So these are cells that are constitutively expressing green fluorescent protein in their cytoplasm. So they're always lit up, and he dispersed them throughout the pore space at a very dilute volume fraction so they don't interact with each other. The cells are constantly trying to swim, and we just watch them. And the question is, how is their run and tumble motility changed when they now inhabit this crowded pore space? And so here are some of the first movies that Tapa took when we did this experiment. Okay, these are two separate cells. I just put the cells on the same slide, right? These movies will loop. they're moving, you're watching the cell body as these cells move through these crowded pore spaces. So there's obstructions everywhere. And when Tapa showed me these movies, I was flabbergasted, right? I fell out of my chair when I saw these movies because even though these cells are trying to do run and tumble motion, right? The pores are big enough, we're not squishing them, right? This mode of motility, it doesn't look like they're doing run and tumble motion. Because remember, in run and tumble motion, the tumbles are effectively instantaneous. A cell does a run and then another run, so on. Here, it looks like these cells, they hop through the pore space over a much shorter length scale until they get stuck. And then the cell wiggles around in a tight spot of the pore space for quite a while before it can get out, pop through the pore space, and get stuck again. They're intermittently being trapped in tight spots of the pore space. And so this is a distinct mode of motility that we call hopping and trapping. And so we've characterized this mode of motility extensively by tracking the cells in this 3D pore space. The details are in these two papers. We've looked at the various statistical descriptors. I won't go into the details in the interest of time. For example, the mean square displacements at intermediate times are subdiffusive, and then at long times they become diffusive. We can characterize various aspects of that motility. We've shown that the hopping lengths are set by the length scales of the straight line paths that fit in the pore space. It's set by the geometry of the pore space. We can look at not just the cell bodies, but also the flagella. So here's another movie where you can see the flagella that are labeled in magenta. This movie will loop again. I love this movie. I think it's very insightful. This poor little cell is initially trying to move to the left. You see the flagella are bundled. They're spinning around. They're trying to push the cell to the left. But you'll notice, as you can see here, it doesn't go anywhere. The cell body's wiggling around due to that torque. The flagella actually remain bundled for a really long time, 11 seconds, much longer than the typical 2 seconds in bulk liquid. And then the cell finds just the right orientation at the right moment for the flagella to unbundle, rebundle on the other side, and then the cell keeps hopping through the pore space. And we were able to,
Sorry to
yep,
interrupt you, Sujit, again.
go
There
ahead.
is a technical question from
Yep,
Francesco. He's asking, this is a confocal plane. How do you deal with
right.
the four direction?
Great question. So this is a three-dimensional experiment. These cells are moving in 3D. They're away from, the boundaries of the experiment. We make these quite large, but we're just looking at a 2D slice through a 3D plane, a 3D volume. And so that's a good question. And so we've done a lot of analysis to verify that the errors in our tracking, the fact that we're missing the third dimension, are minimal. And also, we've done experiments where we kind of change the depth to which we image, as well as tracked cells moving in 3D by adjusting the focal plane. And we found minimal differences between the data in all of those cases. But that's something that's really important to keep in mind when doing the analysis. So that's a great question.
I
Thank
have
you.
one question.
Oh boy.
My gosh.
Dutta, I think you have to adjust your microphone.
Yeah, somehow.
You can switch off your speakers in the meanwhile.
My
Okay, maybe you can try to write it on the chat and then we read it.
I don't know.
Okay, Sujit, I think you can go on and while Dutta will
Can you
write it
hear
on the
me
chat.
now properly?
Now yes, yeah, please.
Because I was trying to use my desktop and I don't know, some echo. And my question is, so here in the simulation, you are trying to use the Hamiltonian approach, and then
Sorry, just to clarify, these are experiments, not simulations.
okay, so how you are giving the data that entropic traps, and how do you know that? We know from physics that it's an entropic trap because free energy plays a very important role in navigating any living cell or moving of a cell which are interacting, or in a media where interacting particles are, try to habitate or move some. So how do you decide this is is a hopping, trapping? This is just random based on random walk, cell sorting, or has the thermodynamics?
Okay, yeah, great question. So I wasn't going to go into detail about that actually, because there's many other things I was planning on sharing, but I'm happy to talk more about it afterwards. So what we do is we track the individual cells, and for example, so we looked at the distribution of the trapping durations, and there are characteristic predictions for what those trapping duration distributions look like when you have particles, as you were saying. Typically people think about thermally activated passive particles moving through disordered energy landscapes. And what we did is by tracking these cells and measuring those trapping durations, there are characteristic power law tails that you can see in those data that are reflective of these models that people use to study how activated particles move through landscapes of entropic traps. And so we put the details behind that in these two papers. I encourage you to take a look at those if you want to learn more about those details. But I'm also happy to elaborate more afterwards if you're interested.
Okay,
But
sure.
that's a great question. And yeah, it's interesting. We found that the way these things move through 3D porous media, you can think about it as being like active particles moving through disordered free energy landscapes, where now the tight spots of the porous medium are like entropic traps that reduce the number of configurations that the cell can take. And I can elaborate more on the details
That
of that.
depends on so you have given some degrees of freedom. The degrees of freedom is being given to the particles, each particle in the.
So the degrees of freedom they have are the different orientations that the cell body can take. Exactly.
Okay, maybe we can elaborate later.
Yeah, I'm very interested because I don't use on bacterial E. coli cell, but the similar in human cell system, tissue system, it's a confined but not porous. Only that we allow diffusion. Yeah, if the system, after stabilization, it is a requirement for the stabilization or equilibrium requirement of the system. But how with the cell reorient jumps, we all do is based on either you can use a Hamiltonian approach or a Lagrangian approach, the action principle. So that's why continuum model I do, but I'm very interested to have your paper to look at it more carefully. So how you do it. Thank you so much.
Okay, great. Yeah, I think you'll probably find these two papers very interesting then. And I encourage you to take a look. I'll move on just in the interest of time, if that's okay, Marco.
Yeah, we have a very quick question actually that probably is very easy to answer. If you observe any loss of flagella in the crowded environment.
Any loss of flagella? Oh,
Yeah.
Great question. We haven't seen that. No, not unless they're starved, right? So when they become starved or stressed out, for example, you hit them with antibiotics, then yes, there are cases where they do lose their flagella. But in these experiments, no. They retain their motility. They keep going. Great question. And so, long story short, and again, this relates to the several questions that were already asked. By tracking these cells, we were actually able to come up with a revised prediction for what the long-time diffusion coefficient of these cells should be as they move through these crowded spaces, given the characteristic length and timescales of this hopping and trapping process. Again, I won't go into the details in the interest of time, but we were able to show that revised long-time diffusion coefficient works much better at capturing these long-time dynamics in crowded media. And we also generalized these results further through simulations of active polymers moving through 2D and 3D porous media, where again, we found that they exhibit these hopping and trapping dynamics. And actually, by analyzing the hopping and trapping, we're able to come up with a geometric criterion at which this diffusion coefficient is optimized. And I'm happy to chat more about that afterwards if you're interested. But actually, I'm going to move on to the main topic of today's talk, which is collectives. So everything I told you about is for individual cells, right? Moving around and not interacting with each other. But in nature and many applications, we care about groups, about collectives of cells that can potentially interact with each other. And so the next question that we had is, how do multicellular collectives spread through crowded porous media? And to do those experiments carefully, okay, is actually a little challenging. You have to be able to define where the cells are very carefully in 3D. And we realized that this hydrogel platform that we came up with actually gives us a very nice way of doing that. In fact, we figured out a way to use these hydrogels to 3D print microbial communities, to 3D print microbiomes. And so if you have a given microbiome of a given composition and 3D structure, chances are we can design that for you. And the idea is very simple. Again, I told you these granular hydrogel matrices are like ball pits for the cells, right? And so, just like a ball pit or a granular packing, if you go in and you push, something through that ball pit, what you do is you arrange the balls locally, you locally yield that packing, and then they just repack, right? So what we did is we built a 3D printer in my lab where you have an injection micronozzle that goes into this hydrogel ball pit. It extrudes cells from the tip. It traces any 3D path that you want. As it moves, it pushes the hydrogel grains out of the way, and then they repack around the cells that you've introduced in place and hold them in place, right? So the hydrogel acts as a support medium to hold the cells wherever you want them. So you can truly 3D print whatever you want. Not only that, you can go back in and take things out afterwards if you want to assay them offline. And so, if you did this with a bulk cross-linked gel, which is typically what people do, right? If you move the nozzle through, you will irreversibly break bonds. But here, you're just pushing hydrogel grains out of the way. It's a self-healing yield stress medium. And so you can 3D print any shape you want. We were inspired by similar approaches people have come up with to 3D print tissues and organoids. We've now hacked this to 3D print microbiomes. And so, of course, we had to 3D print the Princeton shield. The different colors are different layers at different depths. Because I was at Princeton for a long time doing this work before I moved to Caltech. We're currently working on 3D printing the Caltech logo. We're having a lot of fun engineering all sorts of designer microbiomes, but for the purposes of studying collective spreading, we just 3D printed the simplest possible geometry that you can imagine. So this, what you're seeing here, is a cylinder of densely packed E. coli embedded within one of these hydrogel matrices. So the cylinder is much longer than what you see here. This is just a small field of view. The cylinder is embedded and completely surrounded by this porous granular hydrogel. We do 3D imaging, so you're looking at a slice through the midplane. It's pretty zoomed out. Each pixel is a single cell. The entire width of your screen is several thousands of cell body lengths, right? And so the question is, how is this initial distribution of cells going to spread through their surroundings? And so I actually want to do an audience poll. Any guesses? How is this initial distribution of cells going to spread through their surroundings?
Some guess from the audience?
Um,
Maybe
I,
the,
I I
maybe
guess
the flat plane?
there will be some radial distribution because the it will be axial more
Okay.
axial
And
more. That
Rosalind,
is the only path of
okay.
growing.
Rosalind, you had a thought?
It could be flat.
Okay. Okay. Very nice. Okay. These are great guesses. I'll tell you what I thought. So, I told you by tracking the individual cells, we found that at long times it's a diffusive process, right? You can think about it diffusively. So I thought that this is just going to diffusively spread, right? This will be a nice diffusive problem. I was wrong, and Rosalind was right. Here's what we saw. So again, this movie will loop. They self-organize into these fronts that coherently move at constant speeds over incredibly large distances, right? These are thousands of cell body lengths. Somehow they're coordinating their behavior. Again, this is an end-on view because we do 3D imaging. You can see it truly expands radially outward in these coherent fronts that just move at a constant speed over incredibly large distances, right? We
Can I
can
make a comment? Is this possible?
Sure, go ahead.
That's because basically I think it's the telegraph equation that describes the thing, not the diffusion equation, is it? Because it's a persistent random walk, not a...
Great point, and hold on to that thought for one slide.
Okay.
Yes, but you're absolutely right. And so we can just zoom in and track the individual cells. You can see that they're still doing this hopping and trapping, but as I think Francesco just pointed out, there's a slight bias, right? And that enables them to move as a group, even though they're not secreting any signals to each other. They can't see or hear each other, right? Somehow they're coordinating their behavior as a group. By the way, by tracking the individual cells, right, we were able to find that something interesting about biology. It turns out that the mechanism they use to bias their motion is very different in these crowded media compared to in bulk liquids. And I can chat more about that afterwards if you're interested. But what's going on? How are these cells coordinating their behavior to travel in these groups? It's chemotaxis. And so what's going on? Well, so here's our cartoon of what's going on. The cells are in green in this cylinder. They're surrounded by this nutrient replete matrix. The nutrient is shown in orange. So what do? They consume the nutrient that's locally available to them. They deplete the nutrient, and then they establish this gradient. And as they move, they sense that gradient and they bias their motion up that gradient. That's chemotaxis. As they move up this gradient, there's so many cells in this front that through their collective consumption, they continue to propagate this local nutrient gradient, that they continue to bias their motion up. And so they keep traveling as these groups. These are self-sustaining fronts that just keep moving in these groups as long as there's more nutrient available outward, right? And so we can model this. Very simply, you can write down a simple equation for what the nutrient is doing. It diffuses with a given diffusivity, and it's consumed by the cells at a given rate that's proportional to what the cells are doing. And this function here is it reflects Michaelis-Menten kinetics of uptake. And so this is coupled to what the cells are doing. And so what are the cells doing? Well, the cells have a net flux due to their motility, and over large length and timescales, they also have growth. And the flux has two components. One is a random component that reflects this hopping and trapping diffusion, and the other reflects chemotaxis, where here you can see that chemotaxis, right, is reflected by the spatial gradient of how these cells sense nutrients logarithmically, this function f. And I will get back to that function.
So I'm just confused in one thing. That means
Sorry, I wasn't finished. Let me just finish that real quick, if that's okay. This chemotactic function here, this term is the spatial gradient of how these cells sense logarithmically sense the nutrients, and so the nutrients are described by the variable c, and the function f reflects that logarithmic sensing. And so together, these define what's known as a chemotactic velocity. Yes, there was a question.
okay. I'm confused because you showed the porous, controlled porous cylindrical confinement domain. So the porosity on the wall, on the surface of the cylinder, so that the nutrient can go and move back and forward, right? That's why bacteria has a tendency to move towards the surface, the perimeter or whatever, instead of. So that's you choose how you allow the nutrients to move in and out from the system. Is that correct?
Sorry, I didn't quite understand the question. Yeah.
So you are now showing the orange part, that is the nutrition are diffusing into the system, bacterial domain or in and out. So that is the choice you have given to the system.
That's right. So the initially there's nutrients everywhere. It's completely
Mhm,
uniform. And this
mhm,
connects to the question that was asked before about gradients. The
mhm.
cells, through their consumption of the nutrients, establish that gradient that they
Yeah, they
respond to.
established that. Okay.
Yeah. Thank you for the question. And so, we can write down this model, we can numerically solve it. It works really well in capturing the dynamics of what's seen in bulk liquids. It didn't work so well in capturing what we saw in our experiments. And through lots of work, we realized that, of course, we have to update these transport parameters, the diffusion coefficient and the chemotactic coefficient, to incorporate the influence of confinement. And when we do that properly, and the details are in this paper at the bottom, then we found really nice agreement with our experiments. And here's just an example of a numerical simulation of these fronts showing what we see in the experiments as well. We've also explored what happens when now these cells can secrete autoinducers and change their motility and stop moving when autoinducers exceed a certain threshold and they form a biofilm. And in that case, you get additional dynamics that are really interesting. And we describe those in this paper here. And we've also explored how these chemotactic dynamics influence active phase separation. And I won't go into the details of that in the interest of time, but we put that in this paper here. But okay, this is fine. We can do these experiments. We see these beautiful fronts. We can model them in this continuum description. But something about these experiments really bothered me for a long time. You can see these fronts are moving, they're flat, they're so smooth, even though these cells are constantly being perturbed, right? Some cells move faster, some cells move slower, some cells, they're constantly hitting obstacles, yet these fronts are always flat. And so why is that? How are the cells able to regulate their speeds to keep traveling as a group? I told you that we can 3D print whatever we want. So we intentionally perturbed these populations by putting some cells ahead, some cells behind. We 3D printed squiggles. And what we found in the experiments, no matter the amplitude, no matter the wavelength of the squiggle, we always saw the same thing. They regulate their speeds in this group so that they stabilize these undulations, right? They stabilize these perturbations. The cells at the peaks move slower. The cells at the in the rear move faster, and they catch up. And then they keep traveling as a group. And we see this in the simulations as well. And so what's going on? So through a lot of work, we realized that this population scale feature reflects a simple feature of how the cells sense and transduce external stimuli. So let's go back to this chemotactic velocity that I mentioned. It goes as the spatial gradient of how these cells sense a nutrient stimulus, right? This function F. So typically, people describe this function as just a linear function, right? That's this dashed line here in this plot, right? Typically, people say the chemotactic velocity goes as grad C. That's not strictly correct, right? People have measured these functions. This function that I've shown here is the most widely parameterized function through experiments. But as I'll show you, as we showed, the functional form doesn't actually matter. What matters is that this function saturates. And so this function is shown by the solid line here. And this is a common feature of many living and active systems that sense external stimuli. Your ability to sense an external stimulus doesn't extend over an infinite range, but it's bounded, right? And so here you'll see this function, it rolls over with increasing nutrient concentrations, right? Your ability to sense nutrients saturates at sufficiently large nutrient concentrations. So why is this important? So if we go back to this chemotactic velocity, you can expand that out, right? And in the spirit of linear response theory, right, grad C, the spatial gradient of the nutrients, that's the chemotactic driving force, but it's modulated by the slope of the sensing function, which is like a response function, right? And that diminishes, that decreases as you increasingly oversaturate these cells with nutrients, right? My analogy for this is, I love coffee. Many of you, I'm sure, love coffee. So let's say, right, you you've been drinking coffee all day and it's the afternoon. One of your friends texts you and says, "Hey, this hip new coffee shop opened up down the street. You really have to go." Right? So the driving force is very strong, but you've been drinking espressos all day. You're over caffeinated. Your response function is going to be low. You're not going to go to that coffee shop because you're oversaturated. Now, let's say instead, okay, you pulled an all-nighter in the lab, you wake up early morning, you really need your caffeine. Your response function will be high, but the only thing that's open is the gross gas station down the street that doesn't have very good coffee. If you need your caffeine, the response function will win and you will go there and get your coffee, right? And this response is what dominates this behavior. And we can see that here, this is a simulation showing contours of the equal nutrient concentrations, right? As you might expect, at these peaks, you have a steeper nutrient gradient compared to these valleys, and so you will have a stronger chemotactic forcing at these peaks. If that was the end of the story, these leader cells would move faster, these would move slower, and this population would tear itself apart. But because these cells are jutting out, they're exposed to more nutrients. They're oversaturated. They're out here. Whereas these cells in the rear are hungrier. They're over here. The response is stronger. And that stronger response wins. And when we put this all together mathematically, we can calculate how much faster do these trailing cells move compared to these leading cells. We can compute how long does it take for these cells to catch up. And that time scale is exactly what establishes the time scale over which these large scale perturbations to the population smooth out. And so what we think is
I'm sorry
interesting.
to disturb you. I have one question. So the response function instead of you have described as a logarithmical mentioned kind of description. There's a distribution, right? That if you impose a distribution like Gaussian and Poisson distribution of the nutrition and how the bacterial response function, responds into that. Let's say a life cell, and then you have a distribution of interacting, taking from your neighbor or from the nutrition level, or a little bit realistic more. I don't.
I'm sorry, I didn't really understand the question, but maybe in the interest of time, we can revisit the question at the end of the talk.
Yeah, thank you. I agree.
Yeah, I'd love to better understand your question. But in this paper shown at the bottom here, I want to emphasize that we proved that the actual mathematical form of the function doesn't matter. What matters is the curvature of that function. But I'm happy to chat about that afterwards. What I think is interesting about this is I think this yields a kind of general principle in biophysics that just this simple feature that your ability to sense external stimuli is bounded, which is very generic in many living and active systems, can enable an entire population to stick together and resist perturbations to its morphology over large scales. So I think that's very interesting. And so maybe over in the last few minutes, I'll tell you about another regime of microbial life that you can explore using these experiments. Everything I've told you about so far was for the case where the pores in these packings were big enough for these cells to swim through. And so motility dominates how they spread. But if you make the pores smaller and smaller and smaller, eventually you get to a threshold pore size comparable to the body size, where even if the cells were nominally motile, they're stuck in the packing. They can't swim. And now all they can do is they can consume nutrients, grow, divide, and push their progeny to the next pore. And so this is now the growth-dominated limit. And in fact, confinement in these hydrogel packings gives us a very clean way of testing the relative influence of motility versus growth on how these populations spread. And so when we did this exact same cylinder experiment, right, but now in this growth-dominated limit, we saw something completely different. Remember, we saw these beautiful smooth fronts that moved outward, right? In this growth dominated limit, we saw something completely different. This is what we saw instead. These colonies grew and they formed broccoli, right? They formed these beautiful multi-scale fractal-like structures. We can characterize the structures down to the single cell scale, right? And indeed, they are fractals at these small length scales. To use the analogy of broccoli, you can see there's a characteristic floret size around 200 micrometers that we consistently saw over and over and over again. And so we tested this with different E. coli mutants. We saw the same thing. We saw the same thing with different hydrogel packings of different sizes and stiffnesses and so on. We tested different Vibrio cholera mutants of different biofilm EPS compositions. Same thing. We tested Pseudomonas aeruginosa, which is another biofilm former. Same thing. We tested a strain of bacteria that's found in kombucha because I really like kombucha. We saw the same thing. This seems to be a generic feature of how microbial colonies, dense microbial colonies, grow in 3D. And it turns out that the reason why this happens is due to a fundamental difference between growing in 3D versus growing on a 2D surface. And this is revealed to us actually by work that Rosalind did earlier relating to the influence of nutrient depletion. So imagine that you're a microbial colony growing on a 2D surface, right? You always have access to nutrients from the third dimension. And so if you are in a nutrient replete conditions, you'll just keep growing outward, right, as a closed pack colony. But now imagine you're a dense microbial colony growing in 3D. Even if you're in nutrient replete conditions, at some threshold size, if the colony is dense enough, there will be some growth limiting substrate that can't penetrate throughout the colony because the cells on the outside have consumed all of that nutrient. And you can estimate very simply how far a growth limiting substrate will penetrate into one of these colonies just by comparing diffusion with consumption, right? And in these dense colonies, it's actually not that far. It's just 10 microns. That's just a few cell layers deep. And in fact, we can verify that with imaging, metabolic activity is just localized to this thin surface layer. And so random fluctuations in growth take over. We constructed a model of these active colonies as active fluids, where growth is coupled to nutrient availability, performed a linear stability analysis of that model, and we found that it yields a most unstable length scale given by approximately 20 times this nutrient penetration length, which is the only other length scale here. And that indeed captures the length scale of these broccoli florets. Moreover, when we took that model, did simulations with some noise, we were able to capture many of these rough fractal-like features that we see in these colonies. More recently, we've started looking at mixtures. For example, here's the same experiment, but now you have purple and green cells well mixed initially, and when you let them grow, they self-sort. They spatially sort themselves into the different florets. We think this reflects a founder effect in 3D, and this is something where now Mira has come up with principles describing, depending on the initial conditions of this colony, what kind of pattern will emerge in a mixed community. And this is something we're continuing to explore because we think it's important to maintaining genetic diversity in mixed populations. We've also explored the shapes of the interfaces between different cell types in mixed colonies, and we found that you again can get these rough fingering patterns, and we adapted our active fluid model for these interfaces, and we found that we can again capture the shapes of these interfaces using this simple model, and the details are in this paper shown here. And so with that, I hope I've convinced you all that you can observe a lot just by watching, indeed, right? I gave you some vignettes of how we've applied this philosophy to studying microbial populations in different settings. We've been studying microbes in many other contexts as well. And these are the people who've done all the work and who really drive the group forward. Thank you all for listening. Thank you for your questions. And I'd love to continue to answer any questions you might have.
Thank you, Sujit, for the very nice talk. And a lot of questions came up actually. Some of them I already read it from the chat, and the one that are left actually you already answered during the last part of the talk. So probably we don't
Oh,
have new question. Yeah, yeah, exactly. People were anticipating you went after you explained.
great.
So
And I
now
have a
we'll take.
question now.
Just a second, just a second, Dutta. Maybe we can go in order. I would suggest to raise the hands, and Rosalind was actually fourth, so please, Rosalind, go.
Okay, thanks. Thank you for a really nice talk. It was great.
Thank
Um,
you.
I had a question about the last part where you see the surface instability and the
Yep.
roughening transition. That's super nice. I don't think anybody's actually really seen that in an experiment like that. It's really nice. What I was wondering is theoretically, I think even in the 3D geometry, it should be possible to get a flat front depending on the parameters. Like, can you kind of tune, I don't know what exactly, but can you sort of tune maybe the porosity or something and go through a transition from sort of flat to branching?
That's a great question, and I'd love to think more about that. We've never seen that in our experiments. Just to be clear, we've never seen non-rough shapes in our experiments. We've done this. We've even done this with yeast. I mean, this appears to be very generic to microbial growth in 3D. But you're absolutely right. In numerics in the simulations, I think that we can reach regimes where you have flat fronts, particularly when this characteristic penetration length scale, and again, this is inspired by things that you've thought about, is much larger than or of order of or larger than the size of the colony. And so, I imagine that the way to tune that isn't through the porosity of the hydrogel packing, because actually in these experiments, the hydrogel is just serving to hold the cells in 3D. And other than that, it imparts very little mechanical resistance or diffusive impedance to nutrients. I think the way we would do it, but we haven't accessed that regime experimentally yet, is by playing with the diffusivities of the nutrients. So maybe this is what you were alluding to when you mentioned the porosity, or the consumption rates by the cells and the density of the
Yeah,
population. I
I
think that would be the way to do it.
because I thought that would be easier to tune than anything else.
Yeah,
But,
it's true.
yeah,
Yeah,
anyway, yeah, thanks a lot. That was
yeah,
interesting.
it's a good point. We've never seen it, but I think there's a wide parameter space out there to explore.
Thanks.
We had a question from Oliver, but probably Oliver left, or the question was not.
It was basically
Okay.
answered in the previous
Oh,
question.
cool.
Thank you.
Okay. So we have a question in the chat. I was wondering if motility is required for the fractal formation, or it is only driven by growth. Actually, yeah, I was already answering that way, but yeah, cool.
Great question. Growth, only growth. This is purely growth dominated. And that's the interesting thing is, all we did. So in this experiment, you have this cylinder, right? We basically just did the same experiment, but systematically reducing the pore size, right? And when the pores are big enough than the cell body size, if the cells are motile, then it's just motility dominated, and you get these beautiful fronts, right? That I showed you, these smooth chemotactic fronts. Once you get to the threshold pore size that's comparable to the cell body size, even if the cells are motile, but we also did this with non-motile cells, even if the cells are motile, they can't swim. And in that case, you go to the growth dominated regime, and that's when you get these fractal-like structures. So the porosity, or the pore size, I should say, gives us a really nice way of tuning the relative importance of motility versus growth in the population dynamics. And actually, in this paper here, this PLOS computational biology paper, we actually kind of analyzed how the degree of confinement tunes this balance. And there's interesting kind of mathematical features that emerge from thinking about the competition. So thank you for the question.
Probably we have time for a last one or two questions from Dutta, please.
Thank you. Thank you. So I was very interested in the fractal features of this, formation, small scale structure. So how do you decide the diffusion length scale over the confinement length scale for observing such feature? Because you gave here the diffusion, the length scale as a square root of already. So that is the only thing you used, or from the physical system, you are deciding the diffusion length scale and for formation of small scale fractal structure. So
I didn't fully understand your question, but I think your question was, how did we calculate this length scale?
yes, so diffusion length scale is very important in having the fractal form or confinement structure, or very important. Very,
Yeah.
very.
The point is that exactly. And the point is, so, let's say you're a dense microbial colony growing in 3D. In the surroundings, right, there's some growth limiting substrate,
Mhm.
okay, that the cells need to grow. And its only way to get into the colony is by diffusion with a given diffusion coefficient that's d sub s here. And that's just a property of whatever that molecule is, right? Oxygen, nutrients, whatever, right? And so that's just given by the diffusion coefficient of that small molecule in the surroundings. So that's the diffusion coefficient. And so that needs to diffuse into the colony for it to be able to transport it to the cells. But the cells in the colony are taking that molecule up, right? They're consuming it. And so you can calculate, right, how the dynamics of how they consume that nutrient. So here, K sub S is the consumption rate per cell. Here, rho is the density of those cells. C half is a Michaelis-Menten, reflects Michaelis-Menten kinetics of uptake. And so this basically compares, right? This is the length scale over which that diffusing growth limiting substrate has become completely consumed by the cells in this colony. And that's the length scale to which it penetrates into a colony. So that's this characteristic length scale.
Okay, I have one question because I am not an experimentalist, but I'm just curious. I saw that you applied for patent. So what is the reason for patent application? And is it possible that the human tumor cell, you can confine the cells with similar idea, giving food localized and trapping the cells as a treatment or as a drug delivery, or so what is the reason for patent application here? How it is related to?
Sure. So thanks for the question. So we have some IP on this, and the reason is because, everything I told you about here is for very fundamental studies of how these cells behave in 3D. And I mentioned how we developed this way to 3D print microbial colonies. And so here's just a movie showing how this is done, a version of how this is done. We're excited about this because, microbial colonies, especially structured microbial colonies, are important in many applications in the body, for example, as probiotics, in the environment for bioremediation, right? And people often know what kinds of species or strains are useful, but they need to be arranged in a very precise 3D spatial structure for them to be able to work properly. And our experimental platform, even though our goal was to do fundamental research, it now gives us a way to design those 3D communities. And so we're excited about the practical applied potential of this approach to essentially 3D print designer microbial colonies for whatever application you want. So that's the reason why we have some IP on this and interest in this, both from an applied perspective. You also asked about using this for human cells, I think it was. That's a great question, because a lot of the features of this technology are not specific to just microbes. My lab's focus is just on microbes because we think microbes are fascinating and that's our real focus. However, many other groups, and I listed some of those groups here, have used similar approaches to 3D print mammalian cells, tissues, organoids, and so on. We were inspired by those groups. In fact, my former postdoc Tapa, who's the one who initially came up with this, his surname is Bhattacharjee. He's a faculty member at the NCBS in India, and that's one of the things that his group is really pioneering, is using this approach to study mammalian cells as well. And so if you're interested in that, I encourage you to check out his work as well as these groups here. My group is really focusing on using this to study microbes because we think microbes are just fascinating.
Okay, well, we have really the last question from Caroline, please.
Yeah, just to say, really great talk. One quick question. So now those cells do not aggregate. I think E. coli doesn't aggregate. So what happens when they're allowed to aggregate?
Ooh, great question. So actually, interestingly, E. coli can aggregate under starvation conditions and so on. Howard Berg studied that a while ago. What happens when they aggregate? I guess that's a broad question. There's many ways I can answer that. Maybe I'll just highlight one recent study we did of kind of related to your question. My student Sebastian did some experiments recently studying how E. coli, growing immotile E. coli, aggregate in polymeric solutions, okay, where now they aggregate due to the influence of surrounding polymers. And in that case, and I'll just show you one movie, which I think is just really striking. So here, I'll share my screen again. And this is something that I wish I could have talked about today, but I just didn't in the interest of time. So here, Sebastian studied E. coli growing in polymer solutions, for example, in mucus, because we're interested in how these things grow in the body. And in that case, he saw something really cool. Okay, so here's what we saw. So the green are E. coli, but now they're growing in a polymeric solution. So they're aggregating together. And look at this. They grow as clusters, but they grow in these beautiful long cable-like structures, right? They have pneumatic alignment internally. They grow, they elongate, they buckle, they intertwine. They do all sorts of cool things. Here's a 3D rendering of this when Sebastian studied this across different aggregation conditions and so on, and actually was able to develop a mathematical model describing the underlying physics, the physics underlying this process. If you're interested in that, please check out this paper here, which the preprint is on the bioRxiv that Sebastian did studying this form of aggregation. So I hope that starts to address your question.
Thanks. Very, very interesting. We don't really observe that in mucus, but yeah, we maybe haven't yet looked enough. I mean, in the codon. Well, nobody knows how the codon looks, but yeah. Anyways, mucus beads. Yeah.
I
Okay.
I would love to hear what observations you've made and see what things where we can connect.
Thank you. Thank you very much.
I actually think we don't have time anymore because we are we have other question actually that people wanted to ask, but I think we have to stop it here. So I would like to thank again Sujit for being here, all the speaker of this year because it's the last one, this one, and we'll start again probably from mid-February, hopefully next year, and the audience again. So that's everything. Have a nice weekend, and thank you again for the very nice talk, Sujit, and the very interactive discussion in the end.
Thank you for having me. Thank you for the discussions. Happy New Year, everyone, or see you next year.
Exactly. Thank you again.
Take care.
Bye-bye. Bye.
Bye.
Nice weekend.